célèbre London School of Hygiene Tropical Medicine et qui a réalisé certains des travaux majeurs de séquençage en microbiologie qui ont éclairé différents pathogènes, comme nous l'a expliqué hier. Ne se contente pas d'un microbe, mais de vue générale sur différents micro-organismes. Et donc il va nous présenter ce midi un aspect particulier dérivé du génome, qui est les aspects de glycosylation chez les micro-organismes et leur éventuelle implication pour les stratégies de vaccins. On le remercie d'avoir accepté de venir nous donner cette présentation aujourd'hui.
Merci, Michel. I apologise, I will have to give this in English. If I gave it in French, you definitely would not understand it. So, anyways. So, first I would like to start off by thanking Michel and Didier for inviting me to Marseille. I've known about their work for many, many years, and it's a real pleasure and honour for me to speak here today. And you know, Didier is an outstanding microbiologist, and Michel, phenomenal output in terms of publications. Although Didier, I have to admit, I haven't read all of your publications. I've read most of them, but not all of them. Anyway, so I thought I'd take this opportunity to share with you exciting areas of research in bacteria, and I'll take you through the journey. It's been a voyage of discovery for us in terms of realising that bacteria, virtually all bacterial pathogens, modify their proteins by glycosylation. So it's, traditionally when we talk microbiology when we were young, it's a thing that prokaryotes didn't do, but like many rules, they're not true and they're there to be broken, and I think we should always question rules, and this is an example where I'll show that for sure bacteria modify their proteins, they glycosylate, and it quite often is important in terms of pathogenesis for bacterial pathogens. So my journey in this area, I'll go back to the beginning, just to explain, to remind you that protein glycosylation is vitally important. You know, 80% of human proteins are modified by the addition of sugar structures. And they play an important role in many biological processes, from conception, from when sperm cells meet the eggs, to death. So I guess that just about covers everything. So the interactions between glycoproteins is very important in terms of basic biological phenomena. And they're quite complex and difficult to study in eukaryotes. But as I mentioned, they are found in bacteria. But the glycocode itself is poorly understood. And another key thing is, in contrast to the cloning revolution, where we could clone DNA for proteins, glycoproteins have really escaped biotechnological applications. So again, in this talk, I hope to explain how we feel that we've overcome that and we're able to make glycoproteins in E. coli now. So going back to my first study of this, it goes back over 20 years, about 1992. I was a young lecturer and I worked on Campylobacter jejuni. It's a leading cause of foodborne disease. And we're studying, and it's difficult to study, we had trouble mutating genes, and I proposed at a meeting it'd be easier to sequence the whole genome than study one gene at a time. And at the time, most people were very unhappy with that suggestion. They said, you can't do that, you take all the money away from our basic research. But I carried on saying that this is a strategically important area. So a few years later, I was lucky enough to meet this man here, who I think you'll recognise, Craig Venter, in 1995. So this was before he published the Homophilus genome, and he came to the UK and he explained about how, you know, we could shatter bacterial genomes, put the pieces together to sequence them. So I went back to my lab and I made a shotgun library of my pet organism at the time, Campylobacter jejuni, and cloned that into PUC and amplified up many clones. And then I started to sequence some of these clones in the old traditional way, and I sequenced 100 of these. It took quite a long time to do that back in 1995. And to my surprise, a lot of the proteins that came out didn't appear to be Campylobacter-like. For example, there was a capsule there, and it was considered Campylobacter doesn't have a capsule. There were other genes there that looked very strange, and there were some that looked like a glycosylation pathway that looked eukaryotic, and I was beginning to wonder whether I had not prepared the DNA properly and whether I'd made a mixed library here. However, there were some genes that did look like Campylobacter, and we carried on. So, moving on 5 years, with the Standard Institute at the time, we were able to sequence completely the Campylobacter jejuni genome. The yellow one is Helicobacter that we were involved in at the time, Helicobacter pylori. And there were surprises, so we were interested. I mentioned about the capsule, and sure enough there's a locus here. It took a lot of persuading the Campylobacter community that Campylobacter had a capsule. And one person said, Brendan, I've worked on Campylobacter 20 years, don't you think I'd know I have a capsule by now? But when we mutated this locus, and when we stained with Alcium blue, we noticed that the layer on the outside disappeared. So for sure there's a capsule, but that's a different story. I'm not going to talk about that today. That also proved to be the penoserotyping antigen. In contrast, people thought it was lipooligosaccharide. Also, the flagella is modified in many ways with different glycans, and that was known about before the genome sequence. However, what's particularly interesting was that the second glycosylation system, a so-called N-linked one, where the asparagine is modified rather than the threonine on an O-linked glycosylation system. So we became interested in this, and so this is the sequence, part of the sequence here, and you can see it's present in eukaryotes and archaea, never been found before in bacteria. So this is the key gene here, the so-called oligosaccharide transferase, or OTAs. So this is the key enzyme that transfers the whole sugar molecule onto the protein. And up and downstream, there are other genes that look like glycosyl transferases. So it looked like a locus, a general glycosylation locus. So we were intrigued by this. So to study it further, in traditional manner, we made a mutant. So we mutated the key oligosaccharide transferase, which is called PIGL-B. So we mutated it, and we compared a proteome extract, that's the wild type, and a mutant. And you can clearly see many of the spots have been lost. Typical of a glycoprotein, the trainer spots is lost in the mutant. So this again suggested this was a general glycosylation system, and we were able to subsequently show it modified over 100 Campylobacter proteins. So we're interested in studying this system in more detail, and what we wanted to do was to say, we know it has a big glycostructure, we want to know what the structure was. So we tried to purify this from Campylobacter, but it's very difficult. It's a difficult organism to grow, and we couldn't get enough glycan for structural analysis. So then a very naive experiment was thought up. We didn't think it would work, but we thought, well, what would happen if we put the whole locus into E. coli, and then also put one of the glycoproteins from Campylobacter into E. coli? Could we persuade E. coli to modify the protein and use the machinery? And in effect, we'll have cloned the glycoprotein, and then we'll be able to purify enough of that to do the structural analysis. So we really didn't think this worked. It's a stupid student experiment, but to our surprise, shown here, we did get a product. In fact, it modifies, 2 bands here, it modifies the PEP3, which is the reporter protein, from Campylobacter in 2 places. So this is demonstration. This is antisera to the glycan, and also soybean-agglutinin electing to prove that we had transferred this whole system into E. coli, and we were able to purify it and determine the structure. So it's actually, this is the structure here. It's heptasaccharide sugar, has an unusual bacillosamine sugar here that adds it to the protein, and then there's 5 more sugars and a branch glucose here. And then we went back, because we had the whole system within E. coli, we thought we'd mutate each gene. So this wasn't too difficult to do. We fired in lots of transposons into the locus in E. coli. So we fired transposons into the plasmid that carried this locus. And then we're able to isolate each of these mutants, shown here, and they produce the glycan, and then directly look at the structure. And you can see that the sugar is being built up depending on how deep you make the mutation into the locus. So this is quite useful, and again, to cut a long story short, it's enabled us to work out exactly what's happening in this general glycosylation pathway in Campylobacter. So this is the locus shown here. So essentially the first three genes here, which are dehydratase, aminotransferase, and acetyltransferase, they convert the hexanac here into the bacillosame sugar, and the next gene adds it on to the undecyl phenophosphate, and then PIGL-AX, PIGL-J and H, PIGL-I adds the glucose here, and then two unusual things happen. First of all, PIGL-K flips the glycan across the cytoplasmic membrane into the periplasm, and then the magic coupling enzyme, PIGL-B, the oligosaccharide transferase, adds the glycan onto the protein here. So that's very useful for working out the pathway. And it was also useful to do comparisons to the eukaryotic equivalent. So you see here the pathway I just mentioned about the sugar build-up, flipping across, and then the oligosaccharide transferase acts. Well, in the eukaryotic cell, if you look at the endoplasmic reticulum, you get the sugar build-up, flips across the membrane. Something slightly different happens here. You get more sugars added, and then the OTA's equivalent adds the sugar onto the protein. And also, I did mention in Campylobacter there are two glycosylation systems. The flagella is modified as well. I'm not going to mention that story. That's an Olin glycosylation system. But again, they're parallels to the eukaryotic system. So we're able to use this to really justify studying these systems in a simple bacteria where you can make mutants, you can mutate this pathway, but it's a lot more difficult to do that in human or eukaryotic pathways. So moving on the story, as I say, we never knew where this genome sequence project would lead us, and indirectly it led us to be able to produce recombinant glycoproteins in E. coli. And then we thought about the possibility of exploiting this. We know that glycoconjugate vaccines are very useful vaccines, and would we be able to use this technology to perhaps make more efficiently glycoconjugate vaccines? So we call this technology protein-glycan coupling technology. And to summarise it, as shown before, it's quite simple really. Within E. coli, we have three different plasmids. We have the plasmid that has the protein, an acceptor protein, and then if it's got the N for N-linked, an appropriate sequon, you can persuade the protein to accept a glycan. So this glycan, it doesn't have to be the glycoprotein like the heterosaccharide from Campylobacter I mentioned. It can be other glycans like capsules or O antigens. So this is what is discovered in the last 10 years or so. So then it's becoming very useful in terms of glycoconjugate vaccine. So to couple the glycan to the protein, we use this, I call it the magic coupling enzyme, PIGL-B. So the theory here is that you put all three plasmids into E. coli, and in a one-step process, we can produce the glycoconjugate vaccine. So we've been working on that principle for the last 10 years, and so is a vaccine company as well, and we have had some success that I will mention now. But I think it's important to mention that the importance of glycoconjugate-based vaccines, because the advantage of having a protein and a glycan is you get a T-cell independent and it's a T-cell dependent response. So therefore you get a longer lasting memory in terms of the vaccine. And another point is that they are quite suitable for children as well. So particularly, it's been used a lot. These are 3 main examples where they've been used and been shown to be very useful worldwide. But sometimes they can't be used in children. And there are problems with some of these vaccines, like the strep pneumonia vaccine. There are over 90 different capsules. So really, the vaccine's only against some serotypes. And that's a problem. You get capsule switching, etc. So the WHO a couple of years ago recommended that new bacterial vaccines, if possible, there is real benefit to having glycoconjugate-based vaccines. But the problem with current vaccines that are made, and they are very successful vaccines, you know, millions, in fact all of us here probably had a glycoconjugate vaccine, is their production. So it's a multi-step procedure at the moment. It's at least 2 fermentation runs, purification steps. It's done by chemical coupling. You have to handle the dangerous organism, purify the capsule or the lip oligosaccharide, and it's really quite a complicated process, which is why these vaccines are relatively expensive. So as I mentioned, what we're proposing is that we could do this in E. coli in a 1-step process. So that's all very fine. So I'm going to take you in the next few slides through 10 years of work to refine this technology. I'm not going to go into great detail, but I'll mention some of the problems. So one of the problems with the Campylobacter pygmy is that it has some specificity. In other words, it won't accept all glycans. It prefers them that have a hexnac at the end or galnac, which is quite a lot of important glycans, but not all of them. So since we obviously identified this in a genome sequence many years ago, many more bacteria have been sequenced, and these pygmy loci are found in the epsilon proteobacteria. So other Campylobacter species have them, some Helicobacter species have them, but most epsilon proteobacteria are actually found in deep sea vents. So if you look here, the majority of the bacteria. So in our very human-centric view of bacteria, the epsilon proteobacteria represent a tiny piece of the cake. But actually, they are found obviously more widely in the environment, particularly in the harsh environment of deep sea vents. So we've been characterising the pygmy equivalents from many other bacteria, and they do seem to have slightly different specificity. So this is useful for building up the glyco toolbox, because what we want to do is have a situation where we could add any glycan to any protein and put this into E. coli to produce recombinant glycoproteins. So that's on its way. The second area we need to develop is we are used to seeing blocks of genes within pathogenic bacteria for capsules, lip oligosaccharides, and for glycosylation systems. We don't really know what all the genes do or what the structure of the glycan is. So over the years we've been able to come up with a quick way of going from sequence data here. So this is Campylobacter jejuni I mentioned. These are many other Campylobacter species. They have the conserved pygmy and they have various other glycan structures. You can tell by looking at the DNA information. So we've been able to do an in vitro assay to first of all test whether you can get any glycosylation, and then if that works we can go directly from that with mass spec to work out what the overall structure is. So we can quickly go from gene sequence to structure, and we believe this is quite useful for synthetic glycobiology, where you have cassettes of genes that will relate to an overall structure. So again, that's quite rapid advance in the last 10 years. So one of the difficulties with what I'm mentioning is, well, what about the protein? Can any protein accept the glycan? They really do need to have a specific sequence that needs to be exposed in an outer loop of the structure. So that created some difficulty because we always had to engineer in the sequence, and it didn't always work. However, more recently in Michael DeLisa's lab, they've been able to show that actually you can just put the appropriate sequence at the end of a protein or glycotags, and the glycan will live. So rather than trying to work out the 3D structure and engineer it within the middle, you can actually just add the sequence at the end. So this is very good now. It means that really you can pick any protein you want to modify to add the glycan. So the last one is we'd like to improve the efficiency of pickle beans, an enzyme. It's not a particularly efficient process in the E. coli. And the structure. So we've always wanted to know what the structure is, and it's very difficult. It's a transmembrane protein. It's difficult to determine the structure of those. So recently, it's published in Nature. Finally, we've got the structure, and it's, as you can see, it's a transmembrane protein. And from that, you can see that there's a pocket for the protein and a pocket for the lipid-linked lipid-linked saccharide. And we're beginning to understand the mechanism and the important residues that are important in this key enzyme. So again, we think that will really help in terms of driving the technology. So I'm going to move on now to show some examples of where we started to make some recombinant glycoconjugate vaccines. So these are some that have been done in my lab. So I'll show Francisella tularensis, Burkhardt Malai, strep pneumonia. We hope to improve on the current vaccine, and we've just started some work on Coxiella as well. So the first one of these, I'm sure this is more familiar to you, Francisella tularensis, quite a nasty lethal disease, and there's no current vaccine for it. So in our model, so this is a prototype, if you like, for our system. So with the 3 plasmids, the protein we chose actually was exotoxin A because it's quite a good, for various reasons, it's quite good as adjuvant activity. And we knew that it had a perfect recognition site for the glycan. So the glycan we chose was actually the O antigen for Francisella that we'd worked on previously. And in the 3rd plasmid, we have the Campylobacter piggle B. So the first step is to actually clone the locus into E. coli, and that's not always an easy task. This is the locus encoding the lipopolysaccharide from Francisella, and I had a PhD student work a few years back, so we were able to quite easily put this into E. coli, and you can see here that we did get fluorescence on the surface as antisera to the lipopolysaccharide. So the next step is to add the other plasmids into E. coli. So one of them contains the exotoxin A, the protein carrier, and this is an example where it's not present, and you can see that we do get transfer. So this is a ladder of bands, and this confirms, shown in green here, because this is the antisera to lipopolysaccharide, that we have got transfer of the whole system in E. coli. And this is a silver stain as well to confirm it. So that worked quite well actually, and we can just grow up 10 litres of E. coli, and that will typically produce 50 milligrams of this new glycoconjugate. So the next stage, so this is the very first attempt, so we're quite pleased with that. And with this very first attempt, we put it through mice to see if it protected. So shown here is obviously the mice died very quickly with the wild type strain, and if you try to protect with just the lipopolysaccharide alone, it doesn't make much difference, but once it's coupled to the XOA, we get this quite large protection, and the people who work with were delighted with this. They said it's the best vaccine that they've tested to date, and we confirmed that it did have a Th1 dependent response. So we're working up the next generation of these, so we can start to mix and match. So this is one of the great advantages of this technology. So I mentioned about exotoxin A. The one we used just had a single recognition site in the middle of the protein. So now we started to tag them at the end, and we could put multiple glycans in there. So we tried to test that. And that seemed to work as well. So we can show here we've got multiple glycans now present on the exotoxin A. This was the one I showed previously, and that's currently going through mice and rats to see how effective it is. But the other area is that we can actually change the protein. We don't need to use exotoxin A. Now, why not use a protein candidate from Francisella itself? So you have a double hit. You have the protein from the organism as well as the glycan. So you have the best of both worlds in terms of trying to drive an appropriate immune response for protection. So we have various candidate Francisella proteins that might make quite good vaccines in their own right, although it's an intercellular pattern, it's quite difficult. So we have a pipeline of these coming out the proteomics studies, and we started to test some of these. So this is DNase-K, GROE-S and TIL1, and it's shown here we have, yes, with glycotase, been able to modify these and produce recombinant vaccines that we are currently testing in mice. So the actual rate-limiting step now is not producing the vaccines, it's actually been able to test these in appropriate animal models. And these are some of the candidate proteins that we're trying to put the O antigen on to really drive a firm immune response. So moving on to another organism that you may be less familiar with, but at my institute, they've worked on Burkholderia pseudomallei for 30 years. So there's a particular problem in northeast Thailand where paddy field workers, they get an abrasion in their skin, they can quite quickly go down with septicemia and die from Burkholderia pseudomallei. Also in Vietnam, and lots of the Vietnam vets from the US many years later came down with latent Burkholderia infection. So again, it's a select agent tier 1 organism. There's no current vaccine, and so we thought we'd try to produce a recombinant glycoconjugate vaccine. So similar to what we mentioned before, this is the locus we're looking at. It's quite a large locus. In this case, the acceptor protein was ACRA. That's one of the compiler bacter acceptor proteins, and the third plasmid would have the piggle B. And this parallels the Francisella work I just mentioned, whereby the first stage we could express the Burkholderia O antigen in this case, and the capsule in fact in E. coli, and we were able to couple it to ACRA, and they've gone through my studies, and again shown here is for Burkholderia anyway a very good protective response, and that was our first attempt. So again, as I mentioned for the Francisella, we're now looking at various protein candidates. So rather than coupling ACRA, which is a compiler bacter protein, we're now looking at these proteins which are from Burkholderia itself. So as I mentioned, we hope to have a best of both worlds where we'll have the protein from the target organism as well as the glycan. So just move on to another example. As I mentioned, there are quite a few very successful strep pneumoniae vaccines used worldwide, and major drug companies have versions of these. But there are 90 different serotypes, and the disease burden is huge, particularly in Africa, where I have a number of students that look at the molecular phylogeny of strep pneumoniae, and they find that we'll have a couple of students that are looking at the vaccine responses to Prevnar, which is the current one that's used that has 10 of these capsules. And they've been able to show that the problem with that is that was made in the US for the developed world, and in some respects that vaccine is actually making matters worse, because some of the more serious serotypes, such as serotype 4, 5 and 1, aren't being encapsulated with this vaccine. So, and apart from the fact it's quite an expensive, relatively expensive vaccine to make. So we started again along the same principles I mentioned. We've chosen. We have 5 capsules here. We're able to express these in E. coli. It's quite a difficult task doing that. They're very big loci, but we've been able to persuade at least these anyway to be expressed, and this is showing it on the surface of E. coli and the appropriate staining as well. So that's good, that's the first stage, but can we transfer this capsule onto the protein? So this protein here is again ACRA, that's our standard one we studied for the Pilobacter, and we have managed to do that, and that's been through mice, and we've got quite good data already on that. But ideally, as I mentioned before, maybe rather than trying to clone all 90 different serotypes from Strep pneumoniae, maybe we don't need to do that. If we have a very good protein candidate from Strep pneumoniae itself, then we could couple some of the capsules to that, and that might give broader protection. So one of the several proteins we're looking at from Strep pneumoniae is deactivated pneumolysin, and we've at least shown proof of principle here that we can produce pneumolysin with some of the capsules coupled to, and again they're being tested in mice. So just to move on about other uses for the technology, you're probably getting the picture now that you can use combinations of proteins and glycans, and you could use that with an attenuated strain. So this is quite an old example of where we didn't put it in E. coli, we put it into Salmonella, and we put in, on page 1 is a Campylobacter candidate protein vaccine, and we're able to glycosylate that. So what we'd like to do now is put this in attenuated Salmonella vaccine strain and have dual protection against Campylobacter. And maybe we could add in less Campylobacter protein and Campylobacter glycan, but potentially we could have a protein from another organism, such as Clostridium perfringens, which is quite a problematic poultry organism, causes enteritis. So these are some other ideas that we're working on. And I'm just going to summarise it here. So we're working on these triple combination poultry vaccines mentioned, and we have funding to work on a triple combination pig vaccine. A company has come out of this. These were the co-discoveries of this in the original science paper. So the PhD student set up the company in Switzerland. They showed some great data recently of coupling capsules from MRSA onto XOA, and that's published in JRD earlier this year. And they have various leads that are going through human trials at the moment, including Shigella and neuropathogenic E. coli. So I'm convinced the technology works. It's taken quite a while to get there, but I think we'll hear a lot more about it in the future. So I just want to finish my presentation by going back to bacterial glycosylation. And as I've alluded to, it's really the rule rather than the exception. So this is just a sample of some recently identified. In a couple of months in 2012, Acinetobacterium has a glycosylation system. Some of the enzymes are shared with the pathways for capsule formation, and it's important in virulence and biofilm formation. There's an O-linked system found in Vibrio cholera and Burkholderia thailandensis. There's an O-linked system found in, I can't even pronounce that, I think it's an oral pathogen. And even more recently, I just picked up some on Staph aureus, glycosyltransferase, and even mycoplasmas as well. Protein glycosylation is found. So I hope I convinced you that more and more of these are being found, and they're being quite invariably found to be quite important in some role in terms of pathogenesis. So even in our own work, we've worked with Clostridium difficile for over 20 years. And we sequenced hundreds or thousands of strains now, and we come up with this picture that there are 5 clades of Clostridium difficile. So the problematic one at the moment, and I think it's problematic here, is clade 2, which is the so-called Briber Type 27 strains, and clade 4, these are the Briber Type 17 strains, toxin A minus B plus. The evolutionarily distantly related ones, clade 5, these are the Briber Type 78 strains that are quite often found in animals and also in fact in young people rather than old people. But there's another one here we've recently identified that causes more severe disease. These are the Briber Type 23s. So the surprise from these was that the S layer shown here, Clostridium difficile has an S layer. Quite often S layers are glycan modified, but they were found not to be in Clostridium difficile. But when we did a lot of sequencing, we found that for sure there was a glycosylation system right in the middle of the S layer proteins, and we're working on that currently to really work out what these genes do in this particular clade. So bacterial glycosylation system, it's popping up everywhere. It doesn't have to be in all strains in your pathogen, it can just be in some. So we're trying to work towards a classification for all of these systems that we're finding. So, sometimes I have the oligosaccharide transferase, shown here. And so we call those OTAs dependent. Sometimes you don't need the oligosaccharide transferase. You can just have a transferase to the flagellin or pili, for example. So in these ones here, you tend to find that they have larger sugars and that they can be general glycosylation pathways. In other words, they modify several proteins. And so we've worked towards this classification here. 4 general classes, O-linked OTAs, so these are typified by Neisseria. The OTAs dependent, independent systems, so for example, I mentioned about the flagellin and Campylobacter. And then there's the N-linked system that I've described, so this is the general glycosylation system in Campylobacter. And recently, what we've discovered that there's another N-linked glycosylation system that's OTAs independent. So this was quite a surprise, discovering yet another N-linked glycosylation system. And it's something that we'd always known that was there. So for example, it adds glucoses and galactose N groups onto the high molecular weight adhesion found in Haemophilus influenzae. So this has been known about for over 20 years, but the significance hadn't quite been realised. And now we realise that this is actually quite a big family of new N-linked glycosylation proteins found in Actinobacillus and Yersinia. So to give you a flavour for what we feel the excitement in this field, these are a number of pathogens that I've worked on within my group. I call them my pet pathogens. And just to summarise, Clostridium difficile, some strains have 2 glycosylation systems. Burkholderia, there are two glycosylation systems. Cholera, Francisella, Yersinia, there's a new N-linked system, and Actinobacillus as well. So, we're actually pursuing these. We're not just trying to mutate these and understand their role in virulence. Obviously, these could be quite useful tools to develop the glycoengineering approaches that I've mentioned. And, let's not forget that glycosylation can happen to the host cell, so it will be a trick that bacteria use. And this was published in Nature recently, where the DEF receptor is modified by EPEC, by this enzyme here that has an N-acetylglucosamine. And even the Clostridium difficile toxin, if you know the mechanism of it, it glucosylates within the nuclei of host cells. Again, I think this is a theme we're going to see a lot more of with bacterial pathogens. So, in conclusion, just to make some statements here, I guess I've been able to describe really the first fully characterised N-linked glycosylation system in a bacterium. I've mentioned about the development of the technology we call protein-glycan coupling technology, and I'm convinced we will be able to produce inexpensive glycoconjugate vaccines. I've touched on the potential about synthetic glycobiology. I think that's another growth area, and as I've mentioned, the discovery of other bacterial glycosylation systems. We're interested in the role in pathogenesis and potential exploitation. So I'm going to finish there. These are various members in my group who are currently there. Most of them are postdocs and past members, and to thank various people. Dennis Linton, who was in my group, is independent now in Manchester. So is Adrian Jarvis as well. We've done a lot of structural work with people at Imperial. Francisella worked with the Defence Science at Porton. Various people at Strep pneumoniae, Tim Mitchell and Jerry Brown at UCL. And also I should mention about the discovery of this was with Marcus Aby at ETZ. Michael Wacker was a PhD student who went on to form Glycan Vaccine, and also University of Alberta, 2 close collaborators, Christian Szymanski and Mario Feldman, were all working on this exciting area. Various funding, and I'd just like to thank you. Thank you very much. Actually, I'm going to turn back from this slide because I realise there's embarrassing photos of me on here. So,
questions? Remarques?
In English, please.
Not in Spanish. So each system is specific for a kind of seed. So each system is specific to have some kind of glycoside proteins.
Yeah.
There is no universal.
Well, I think the transfer enzyme has a common mechanism. So the one I mentioned, Campylobacter jejuni, it does the same thing in other Helicobacter species and in Epsilon proteobacteria. So the enzyme will do the same thing. The adjacent genes will mean that the overall sugar structure will be different, but the mechanism will be the same. And with the new ending system, again, that one is so highly conserved. You know, we believe that it just add a few sugars in the same mechanism. That one happens in the cytoplasm. So with the different four classes I mentioned, they are quite different in the way that they act. But within that class, then you do get the similarities.
I'm looking to see what the viral system. I've not dealt with viruses, so there is a difficult. We could put a viral protein and modify a viral protein. I don't see why I can do that, but we haven't attempted that yet.
Did I understand correctly that in clade 3 of Clostridium difficile there is a part of the apparatus that is lacking? Is that right?
Part of the apparatus
of the glycosylation system. Is it different? Can you come back on the slide? Because what I've seen is that, which is matching very well with my theory, is that so in clade 3, you've lost something. Is that right?
In clade 3, they have this extra locus in the S layer, and this locus of 16 genes is a typical glycosylation system. It's an O-link glycosylation system. So for some reason, the strains in this clade had this glycosylation system to modify the S layer.
What did they lost in your PCR? Those are clade 3.
Oh, sorry, this isn't PCR. This is a protein.
Okay. But what is lost? What is it?
Well, this is the S layers. If you've ever put proteins onto any Clostridium difficile protein, you get two S layer bands. But in the clade 3 here, they're fused together because they have this additional. We believe they had this additional modification. So sorry, I didn't explain that. That's actually a protein gel. Yeah.
Oh,
So the other interesting thing about clade 3...
It's just the glycosylation of the proteins that you were saying here. There is no protein disappearing.
No, not protein. It's actually... I guess it's this band here that's higher molecular weight compared with that one. And the other glycosylation system from the type 27s, the flagella is modified, has an unusual glycosylation system downstream from the flagellin gene. That's also the same as in clade 3, but different in the others. So with Clostridium difficile, you're getting these sets of genes that are specific for certain clades, but they can be present in more than one. And so that was the other observation with the type 23s, that the flagellin glycosylation is the genes downstream are the same as the type 27s.
So it would be interesting to see with you, I'm sorry that Bernard is not here, but Bernard Alascolaiman had worked for 3 years to try to understand neonatal colitis. That is. And what did he find? That he found very, there was preliminary work that are current with it, but what he's finding is that, and what he believes, it's Clostridium botulinum.
Oh, okay.
And they're toxinic as well.
Yeah.
You get the same kind of toxin.
Yeah.
So that would be interesting eventually to discuss with you, because now we're sequencing all the genome to see if these associated with colitis are different glycosylation with you and Bernard, that the odds are not causing.
Yeah, that would be interesting though, yeah.
Because it is very, very significant, but it's not yes/no. It's all the guys with colitis get this, but of course, Metricum, some people without anything get Metricum as well. So
maybe looking at the genome sequences, you'll be able to see similarities.
Yeah, we identify the toxin anyway. So, but we know that there is an hemolysis that is very closely looking to that of this disease. So it makes sense, but it's kids. So because of this job, I think if we go through the same kind of analysis, you may help us to decipher what the problem is.
Yeah. So I think this clade up here, we did another study that we published PNS. It's evolutionary, really distinct 80 million years ago. So I wouldn't be surprised if there were other intermediates that could cause pseudomembranous colitis. I think we've always suspected other Clostridia.
Yeah, it's bizarre. So you mean that clade 3 is mainly associated with Clostridium difficile in children, is that right?
No, not in children. That one's found in patients that have really severe pseudomembranous colitis, toxic megacolon as well. Okay. So there's a big outbreak. And so we actually picked that up by looking at the clinical data. And then we looked back at the strains and we realised they're type 23. And then when we did the phylogeny with MLST and whole genome sequencing, we realised that actually they were new clade. We haven't actually published this yet.
Well, I think this is one of the reasons why I'm doing this team issue with you involved. I really believe that we're at the beginning of a paradigm shift based on genomic analysis, is that we're going to link that all these things that we were saying, well, it's a different clinical form and so on, most of these will be associated with a very specific clone that is giving very specific, clinical form. And I think, well, it's very obvious for Staphylococcus, for example. Yeah. I think it will become very obvious for all the bugs. Yeah. Different clone scopes, different disease, different epidemiology, different target, and so on.
And quite often glycosylation is linked in with some of these differences. I think that's a new thing that we're beginning to understand.
I have two questions. My first one is that what is the biological role of glycosylation in bacteria?
It might take me a long time to answer that one question alone. But this, quite simply, we don't really know. But it does seem to keep coming up. It's been modifying a key determinant within a bacteria, like pili, flagellin, or various proteins. So it gives the bacteria an extra opportunity to potentially move away from the immune system or make that protein. But I think a lot of it, my pet theory, I might be wrong, is I think glycosylation has an important role to stabilise the proteins. In particular, it's known that proteases can often break down proteins. So we've noticed in Campylobacter, for instance, that a lot of the proteins are modified. You can't break them down with endogenous proteases. So bacteria produce a lot of proteases, which are quite often virulence determinants in their own right. So my feeling is that certainly glycosylation plays a big role in terms of stabilising the proteins and prevent proteolysis. And we're trying to prove that, but it's not. It's a nice hypothesis, but it's not that straightforward to prove. But you could ask the same question about glycosylation in human proteins as well. And I guess we don't know for sure, but certainly in terms of stabilising the proteins as they're transported is a general theme that comes out. So anyway, that's my theory about what happens in bacteria. Another question?
The second one is that the presence of the PGNB in the genome of the bacteria, does it mean that bacteria is capable of glycosylating a particular protein?
So if it has a pig or bee gene, does it mean it has a general glycosylation system? Yeah, certainly all the ones we've looked at. So we've looked at about 20 Campylobacter species and Helicobacter species. And in fact, in some of them, they have 2 pig or bees. So we're not certain what the second system does. It's something we're investigating. But if they have that, they have the adjacent genes, which I showed, and then they have that general glycosylation system. But I think the key question is, why is it just some bacteria? Why is it just some Helicobacter species and Campylobacter species that have this particular glycosylation system? I think it's a very ancient system, and the evidence is that most of the bacteria that do have this are from deep sea vents. And again, this is relating to perhaps making sure that the proteins are not to do with stress, and that the proteins are modified such that the stressful environment that they're in doesn't affect the proteins. So bear in mind, Campylobacter, as you know, in a lab, if you grow it, you grow it at a temperature of 42 degrees. That's the optimum growth temperature. That's the optimum growth temperature of avians. So Campylobacter jejuni is not on this planet because of humans. Campylobacter has been here, probably predates humans. It's elegantly evolved to be in the crop of birds, avians, and it's a very ancient, it doesn't cause disease in chickens or birds, it's just a commensal there. But if humans come into accidental contact, then you get severe diarrhea. So the evolutionary driving force for Campylobacter has nothing to do with humans. You know, humans are, like many pathogens, humans are an accidental part of it.
Okay.
I'm around all afternoon. I'm happy to talk about other work, genome-based work as well.
Yes. Once again.